Hey guys, my name's Alan. I'm a tutor here at the Center for Academic Program Support at the University of New Mexico. Today we're going to be talking about some concepts for your genetics class. It's Bio 202. It's the second one you're going to be taking in the intro sequence here at UNM. We're going to specifically be talking about BLAST, which is the Basic Local Alignment Search Tool, and Open Reading Frames, which I've called ORFs here. And essentially BLAST is the Google of genetics. So any publicly funded research on genetics tools or on genes, the researchers are obligated to report their information to BLAST. That information goes in a database and it's got a bunch of different associations with it. And then you, as a researcher in the lab, or as a student here, get to take your sequence that you're interested in, plug it into BLAST, and get some information back out of it on maybe the function or the structure of the gene, right? Open reading frames are the reason that we have things like BLAST. It's not so simple as just looking at a piece of DNA and saying, oh cool, here's where it starts, here's where it stops, there's my protein, and we're done. No, it's a little more complicated than that, and we'll talk about why here. But BLAST uses computers to do that automatically for you. So the first thing we're going to get into is the concept of an open reading frame, right? So just to do a little bit of backtracking on DNA, we know that DNA exists in two anti-parallel strands, right? One of them will go 3' to 5', and then the same one, bonded in hydrogen bonding, with following base pair rules, will go 3' or 5' to 3', right? And our base pair rules are going to be like these. We have A that's doubly bonded to T, and C that's triply bonded to G. And remembering that when we translate this from DNA to RNA, thymine becomes uracil, T becomes U, right? So that's the way DNA looks, and the way DNA codes for a protein is in three base pair codons, right? So when you have a long string of DNA, it's going to be A, C, T, G, whatever. That's going to be read in chunks of 3. So it's going to be ATG, boom, CCG, boom, 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 all the way through, right? And then there's 4 really important codons. Everything is going to code for a protein, except for these 3 stop codons, UAA, UAG, and UGA don't code for an amino acid at all. They code for stop, done, put a tail on it, right? The other important one is the start codon. It's always AUG, and it codes for methionine, which is just a special type of methionine, right? So when we go and we have a chunk of DNA and we want to find out something about it, we're looking for these codons, right? Because all DNA does is it codes for protein. So the trick is, where do you want to start reading from, right? You could find an AUG, but there's only 4 base pairs, and if you have millions of base pairs on a string, you're going to have lots of AUGs and lots of stop codons, lots of things on there. So which one is the right one? And that's where open reading frames come into play. So when you look at a given piece of DNA, there are 6 open reading frames you can look at. It's because 3 base pair codons times 2 strands, the anti-parallel strands, makes 6. 6 different ways you can look at it, right? And we define the open reading frame that's the most likely to carry a protein as the one that has the longest stretch of a coding region, which is what is defined as being in between start and stop. So the longest chain between your start and your stop codon is going to contain your protein. So next we're going to look at a quick sample problem to kind of see how that works in action. This is a good example of a little kind of a sample problem you could get, where you just have one chunk of DNA, one little strand, and you're asked to find the most likely open reading frame to code for a protein. Then once you have that reading frame, you can be asked to write out the amino acid sequence of that protein. This isn't the same thing as a real protein. This is the reason we have BLAST, is because real proteins are thousands of amino acids long. Ours is only going to be a couple. Realistically, you can only do that in an exam, right? So the first thing, the place you'd want to start, is you only have one piece of DNA, and DNA is double-stranded, right? And you know this is DNA because you have T instead of U. So the first thing you want to do is write out the anti-parallel strand. So if this is 5', this is going to be 3', this is 3', this is going to be 5', right? Then we're going to follow our base pairing rules. C binds to G, T binds to A. You just follow that all the way through. Oops. Easy to make mistakes, you can see already. So now that we have both pieces of our DNA, you can kind of see how there's 6 open reading frames. You can start here from this C and run this way. You can start here from this G and run this way. You can start from this T, so you have 6 different ones, right? But always remember, DNA is read from 5' to 3'. So the next thing you'd want to do is kind of look it out and look for your start codons, right? So the first one that I see is right here, looking me right in the face, is this ATG, right? So we'll start there, and this is going to define our reading frame. So I'm going to write it right up here. It's going to be ATG, and I'm just going to put a little line to note that that's the stop of that one. And this is going to be TAA is the next codon. Now that would usually code for an amino acid, but if you come down here, TAA becomes UAA when it's transcribed into RNA, which is one of our stop codons. So that's the end of the sequence we can get starting from that codon. And that's only one amino acid long. It's just methionine. That's obviously not going to be our protein. That's not our most likely sequence, right? And I know it can be tempting to start from this side and go, oh, here's an A, T, G. We could start this way, but that's wrong because this is the 3' to 5' direction. So that's the only one that I see on this strand. So then we can pop down here to our second strand, right? You run through and you look for A, T, G. So here's one right here. And that's backwards, A, T, G. So I'm going to take it and I'm going to write it down here. We're going to go A, T, G, C, G, A, T, C, oh, this is supposed to be a G, C, G, A. That's our stop codon right there, right? UGA, TGA. So then I'm going to write this again. So this is 5', 3'. We're going to write it again. 5' as A, T, G. Leave a little space so you can see your next codon, CGA, and then TGA for stop, right? So this one's a little better. We have 2 amino acids, but let's see if we can do one better than that, right? So that's ATG right there. Rolling through, boom. Here's another ATG right here. So let's erase this strand that we have here. Start again. And by the way, it's totally okay. It doesn't exist in this one because this is in a different reading frame. So this ATG is included, but it's actually this TGA, right? Because it's a different set of 3. But if you were to go AUG, something AUG, that's okay. AUG is codes for methionine. It's okay to have it in your sequence. It's also just noted as your start codon. So this one goes A, T, G, A, C, T, G, T, T, A, C, A, and then T, A, A. That's one of our stop codons right here, UAA. So again, this is 5', this is 3'. Just to make our lives a little bit easier, we're going to rewrite this. 5' goes ATG, right? And then ACT, ATG, ACT, GTT, ACA, and then TAA. And I know that's a little bit of overlap, but you can still see right away, it's immediately obvious. This has 1 amino acid. This one has 2 amino acids. This has 1, 2, 3, 4 amino acids, and then a stop, which does not code for amino acid. So this is our most likely open reading frame to contain a coding sequence for an actual protein. And it happens to be on the strand that you weren't given somewhere in the middle, not nice and easily right at the very front and beginning of a sequence. So it's just an added layer of difficulty when you're looking at DNA to find out where your sequence is. And then once you have this, you can use the genetic code to go and read your sequence to get an actual protein out of it. And DNA is read 5 to 3. Proteins are made N to C. So they have an amino terminus, which is NH2, and it goes all the way to a carboxyl terminus, which is COOH, right? And then they're all going to be in there. First one's going to be methionine. And I don't know the rest of them off the top of my head, so we'll look at our genetic code to get that. The way that we read this code is you start on the left-hand side where it says first position, and you find the first base pair, the first amino or the first nucleotide in your codon. Then you go to the top where it says second position, you find the second nucleotide in your codon. That tells you what row you need to be in and then what column. And then over on the far right-hand side, there's another list where it says third position, and that'll tell you exactly which point in that little area you need to be at. That'll tell you which amino acid your little codon codes for. So in this case, ATG is methionine. It's always that one. Then we have ACT. So if we start over here in the A column, and go over to C, we see that they're all this THR. Doesn't matter what our third position is, and that's fairly common. So this is MET, THR, right? And then our next codon is GTT. We start over here on the bottom. Go over here, T becomes U in RNA. Those are all valine, so VAL, right? And then we go to ACA, start over here in the third row, it's A. Go over to the second position, which is C. Those are all THR. So this is our completed amino acid sequence for our fictional protein. And there you go. The last thing we're going to talk about is BLAST, which again stands for Basic Local Alignment Search Tool. And this is put out by the NCBI, which is the National Center for Bioinformatics, which is a public government resource, right? And as I mentioned, anybody that does genetics research submits their data to their databases, and then other researchers can then use that information to improve our general understanding of genetics and proteomics. So what you could do is if you were in the lab and you have a sequence of DNA or you have a polypeptide sequence you don't know about, you can go and plug it into BLAST, do a search, and get information out. They could tell you, oh, your protein looks a lot like this other protein for saliva in monkeys, and you know you got it from a human, so it's going to be pretty similar to primates, so you say, oh, I probably have a saliva gene here, right? You could do the same thing if you have protein. You could put a polypeptide sequence in, and it'll tell you, oh, this looks like this, and there you go. There's just a few things you're going to need to know about. You're probably not going to be doing a whole lot of searching with this, just kind of a general understanding of what it is and how it works at this stage, right? So when you go to NCBI and go to the BLAST tool, you could select different databases you want to search in. So if you know that you're doing something in a certain kind of organism, you could help narrow it down rather than searching through everything and get better results. Once you do that search, you can go down and you can look, and it will tell you how close you are, how well matched your sequence is, and it'll show you where you have mismatches. And it'll tell you if you have a major mismatch or a little mismatch. So like if you've swapped a polar amino acid side chain for one that's non-polar, that's a big deal. But if you swap a polar one for a different polar one, it's not a big deal. It could be just like a single little mutation or something. So it'll let you know that sort of thing. The other useful piece of information you can get out of this for nucleotides is it'll give you an E value. And an E value works kind of like a P value in statistics where you want a really, really little one. E stands for error, so if you have a really little E value, it's a really low chance that this is due to an error. Should be on the order of like 10 to the negative 7, right? The only other thing that's really useful at this stage is to know that if you were to search for proteins, they could also tell you that this sequence is homologous to other sequences that do certain things. For example, there's a certain type of protein sequence that looks like it spans a membrane. And so if you have a protein, you're not sure what it does or anything about it, they could tell you, oh, this section looks like it goes in a membrane, and you know you have some kind of membrane protein. So it's all about general trends at this stage of the game, nothing too specific. Alright guys, that's all I've got for you today. Thank you so much for watching. Go ahead and subscribe if you want more videos like this. We're going to cover a variety of topics going forward as we're trying to get this thing running. Follow us on YouTube as UNM Caps, Facebook Caps UNM, Twitter, Snapchat, Instagram, the whole thing. Remember, UNM, go Lobos!